Hey everybody, welcome to another lecture video. This time we're going to look at functions of proteins. We're going to start by going a little deeper into what quaternary structure is, and then we'll use hemoglobin and myoglobin to illustrate some examples of the functions that we're kind of interested in. Okay, so the fourth level of protein structure is that of quaternary structure, and it refers to the structures that arise as a result of the interactions between multiple polypeptides. The units can be identical, multiple copies, or they can be different polypeptide chains. A good example is hemoglobin. Adult hemoglobin is a good example of two identical chains and two different polypeptide chains. So let me explain. The quaternary structure is composed of two identical chains called alpha, and then two identical chains called beta. And so the alphas are different polypeptide chains than the betas. In the end, a hemoglobin has 4 subunits altogether. 2 of them are identical to each other, 2 of them are different from the other 2, and yet those other 2 are also identical to each other. So 2 A's and 2 B's. Though the alpha chains are very similar to the beta chains, they do have subtle differences in their primary structure, which would be their amino acid sequence, and so they're not identical. Both of the alpha and beta chains are also related to the single polypeptide chain in the related protein called myoglobin. Myoglobin and hemoglobin have similarity in binding oxygen, but their behavior towards the molecule differs significantly. So notably, hemoglobin's multiple subunits, so multiple subunits being quaternary structure, compared to myoglobin's single subunit, so with no quaternary structure, is what gives these differences. So what I was saying about myoglobin is that myoglobin carries oxygen, let me clarify, it binds oxygen, just like hemoglobin. Hemoglobin is for transport, myoglobin is for storage in the muscles. Hemoglobin is made up of 2 chains, alpha and beta, which are different than the single polypeptide chain that makes up the myoglobin protein. They are all very similar. And so what you can see here in this first image on the left are 2 polypeptide units of a protein with a quaternary structure. So this is the quaternary structure is that there are 2 folded polypeptides, both in their tertiary structures, but together they are the quaternary structure of this functional protein. So of course, quaternary structure describes interactions of individual subunits of a multi-subunit protein. The result of these interactions can give rise to important biological phenomena such as cooperative binding, allosteric effects. We're going to look at some of these. Now, allosteric effects can occur by a series of mechanisms. Allosteric means other site. So enzymes that are proteins that are biologically active usually have active sites. These are the places where the molecules they interact with will bind. Allosteric means that it's a site other than that active site. And those are usually hotspots for regulation. If you want to make a protein go faster or turn off, we can bind things at allosteric sites, which will impose changes into the quaternary structure of the protein, which ultimately changes the active site and then either makes it more receptive to the substrate or less receptive, so it turns them on or turns them off. So a common feature in these mechanisms of allosteric effects is the binding of an effector to the enzyme subunit, causing the enzyme to either transform into its T state or conform into its T state or tight state, which is the less active state, or an R state, which is the more active state. Now, effectors can be enzyme substrates or non-substrates. So homotropic, meaning the molecule that is the effector is also the substrate of that molecule, or heterotropic, meaning the molecule that's the effector is different from the substrate. Now, allosterism gets covered a little later in a different chapter, So right now, that's all I'm going to kind of say about it. So we begin really our consideration of quaternary structure with a discussion of cooperativity, how it arises from the multi-subunit protein hemoglobin, and how its properties contrast with those of the related single subunit protein myoglobin. So we're going to take a look at some cooperativity. Now, there's a video that I tried to embed. This is the one on the right here, that would basically show how these four subunits, the two alphas and the two betas of hemoglobin, this was a movie, when oxygen binds, how they start to change in their conformation. So I don't have that, and it doesn't work. So I'll kind of describe it a little bit. So cooperativity is defined as a phenomenon where binding of one ligand molecule by a protein favors the binding of another molecule of the same type. Now, hemoglobin exhibits cooperativity when the binding of an oxygen molecule by the iron of the heme group in one of the four subunits, and I do have a picture of the heme group while we're talking about it. So it binds one of the heme groups in one of the four subunits, causing a slight conformational change in the subunit. This happens because the iron, the heme iron, is attracted to a histidine side chain, and binding of oxygen lifts the iron along with the histidine ring, also known as an imidazole ring. And so we can't really see the histidine here. I think I have another image. Here we go. So on the left, you can see the histidine is bound to that, or attracted to that that nitrogen, and the iron. That iron. I don't know what I'm saying. It's bound to that iron in the heme core, and then when oxygen binds, the oxygen lifts that iron up, and that in turn pulls on this histidine residue, and so that's going to lead to these little changes that we're talking about. Okay, so it lifts the iron along with the ring, and we get some conformation change. Now, since each hemoglobin subunit interacts and influences the other subunits, they are induced to change shape when the first unit binds an oxygen. And this is described as transfer or turning from the T state to the R state. The T state being the tense or less, that lower affinity for oxygen, to the R state or relaxed, which has a higher affinity. I should kind of write that. So T. And this one has higher O2 affinity. This one has lower O2 affinity. Okay. These shape changes favor each of the remaining subunits binding oxygen as well. And so this is a very important effect in the lungs, where oxygen is being picked up by hemoglobin, because the binding of the first oxygen molecule facilitates the rapid uptake of more oxygen molecules. In the tissues, where oxygen concentration is lower, the oxygen leaves the hemoglobin and the protein flips from the R state back to the T state, which wants to let go of its oxygens. And so it's good at loading it when it needs to and unloading it where it's supposed to. Now, let's see, carbon dioxide is also involved in transport by hemoglobin. Hemoglobin not only takes oxygens to the tissues that you need that need them, but it also removes carbon dioxide. Now, cooperativity is only one of the many fascinating structural aspects of hemoglobin that actually helps the body to receive oxygen where it's needed. It assists in the transport of the product of cellular respiration, which we're going to look at the end of this semester, essentially, which is carbon dioxide. And as that's being produced in the tissues, it takes it to the lungs where it can be exhaled. So like the binding of oxygen to hemoglobin, binding of other molecules to hemoglobin affects its affinity for oxygen. The effect is particularly pronounced when comparing oxygen binding characteristics of hemoglobin's four subunits with the oxygen binding of the related protein myoglobin. So we're going to make comparisons between the two. And so you can see here in this table, we have some different shaped curves. So like hemoglobin, myoglobin contains an iron in its heme group that binds oxygen. The structure of the globin protein in myoglobin is very similar to the structure of the globin in the hemoglobin. And hemoglobin is thought to have evolved from myoglobin in the evolutionary history. As you can see in this curve, the binding of hemoglobin for oxygen is S-shaped, and we call that sigmoidal. Whereas the binding curve for myoglobin, for hemoglobin is hyperbolic. Now, what this tells us is that the hemoglobin affinity for oxygen is low at low concentrations of oxygen, but increases as the oxygen concentration increases. Now, since myoglobin very quickly saturates with oxygen, even under low oxygen conditions, it says that the affinity for oxygen is high, and or it has an affinity for oxygen that's high and it doesn't change. Because myoglobin only has a single subunit, the binding of oxygen by that subunit can't affect other subunits, and since there are no other subunits, consequently there's no cooperativity that we see. Hemoglobin, on the other hand, can exhibit cooperativity, but myoglobin can't. It's worth noting that simply having multiple subunits does not mean cooperativity will exist. Hemoglobin is one protein that exhibits this, but many multi-subunit proteins don't do this. Your book also, now the book has a different image that I should have grabbed, and it shows how the hemoglobin hyperbolic curve can actually be affected under different conditions to be more sigmoidal, and that's a really interesting part of the book. You should definitely find that graph and take a look at it. I'm trying to remember what the effectors are. I think it's pH, 2,3-BPG, and carbon dioxide. Actually, I think I do have that graph in here. So hold on, we'll get there. I think it is in here. Okay, next. What was I going to show about this image here? This is hemoglobin in the presence of oxygen, and hemoglobin in the absence of oxygen. So you can see, presumably this is taken from the same angle, so that it's not just rotated different. We can see actual differences here in the structure. Okay. The lack of ability of myoglobin to adjust its affinity for oxygen according to the oxygen concentration, being low affinity at low oxygen and high affinity at high oxygen like hemoglobin does, means that it's better suited for storing oxygen than for delivering it. And our bodies need oxygen delivered to various tissues for many different reasons. So what we'll see, besides cooperativity, hemoglobin has actually some other structural features that allow it to deliver oxygen precisely where it's needed the most in the body. So this is why we don't see myoglobin as a transport molecule. It's more suited for one job, and that's just holding on to oxygen so that it can be ready when it's needed. Hemoglobin, because of that cooperativity and the subunits, actually gets some different jobs, which is kind of cool. Let's talk about this image I've got here at the top. This is the sequential model of binding. Now, before we sort of get into that exact model, we got to kind of back up 1 second and talk about something else called the Bohr effect. Now, the Bohr effect was first described over 100 years ago by Christian Bohr. He's the father of a famous physicist. I forget which physicist. Now, the Bohr effect, his name is... Come on, how can I forget which famous physicist? Niels Bohr, that's the guy. The Bohr model of the atom. Sorry. So, he's the father of Niels Bohr. Anyways, he showed that hemoglobin's affinity for oxygen decreases as the pH decreases. And so, let's see, which one of my graphs here is showing that? I think that's this one here. So as the pH starts to go down, you see that the saturation of hemoglobin actually, well, I guess it eventually peaks in the lungs, though, sorry, I shouldn't have erased everything that I just erased there. If you pay attention to this little area right here, you do see that in the lungs, the saturation of the blue line is lower than the one of the pink line, right? So there is some difference, at least in the lungs. It's more pronounced in the tissue. In the tissue, you can see the saturation at a pH of 7 versus a saturation of at pH of 7.6 is very, very different. Okay, so he showed this. He was able to graph these things and show this. It's a bigger picture here, though. So he saw also that in addition to the affinity decreasing as the pH decreases, as the concentration of carbon dioxide increases, we also see a drop in the binding. And so here, I think in this other graph, we see the CO2 effects. And so what do we have here? So no CO2, and then increasing the CO2. And so the green line compared to the orange line, an increase in CO2 drops the saturation again. So there are these other effectors. Excuse me. Okay, so binding of protons in carbon dioxide by the amino groups or by some portion of that molecule of hemoglobin changes the structure somehow. And so the model up at the top is called the sequential model of binding. And the sequential model is the only way to help explain what we're seeing here, this hemoglobin's cooperativity. So each of these squares represents a no oxygen bound subunit of hemoglobin. And the circles represent subunits that are bound with the oxygen. And the rounded subunits, or I'm sorry, there's, what did I miss here? There's the square ones, and then there's the circles are bound to oxygen, and then the rounded. So the rounded are kind of these purple ones. The rounded are subunits whose affinity for oxygen increases by interacting with a subunit that has bound oxygen. And so what you can kind of see is that as these subunits change, so I don't know which is the first one, this is the first one to change, it binds oxygen, so then it's bound, and then we see these guys change next, and then they bind, or one of those binds, I guess it's this one binds, and then that affects this one, right? So your book goes into the other alternative. There's the sequential model of binding, and then there's the concerted model. The concerted model says that when one subunit changes, the rest of the subunits change also. And it just kind of goes from, one extreme to the other extreme. The sequential has the differences, these small little increments. And the sequential model is the one that best explains the cooperativity. Now, I'm trying to remember which part of the molecule here actually facilitates. I know it's the side chain, it's the amino side chain, the amino acid side chains in the globin actually helps facilitate these structural changes. Most commonly, the amino acids are affected by protons. Histidine is one of those. Histidine number 146 actually in the beta strand. It's affected by protons, and when this happens, the ionized histidine can form an ionic bond with the side chain of aspartic acid number 94, which has a stabilizing, which has the effect of stabilizing the T state, which is the reduced binding state. And so, to try to understand why an increase in hydrogen concentration, aka a decrease in pH, lowers the affinity, it's because these are stabilizing, or they're creating interactions between amino acids that stabilize the T state. And the T state, remember, is the tense state, and that's the one that reduces oxygen binding. Other histidines in the amines of the amino acids and the alpha chains are also binding sites for protons. Excuse me. Okay, so there's this molecule here. This is 2,3-BPG. We're going to talk about that a little bit too. 2,3. BPG. This is also so biphosphoglycerate. Sorry, I should have said it first and then given you the abbreviation. Biphosphoglycerate. This is a glycolysis intermediate as well that has some good effects. So we're gonna come back to this molecule when we're talking about a little bit of sugar metabolism down the line. All right, another molecule favoring the release of oxygen by hemoglobin. So we talked about how hydrogen binding can do it. We talked about how carbon dioxide, increased carbon dioxide concentrations can do it. Now 2,3-BPG, or sometimes just called BPG. So like the protons in carbon dioxide, this BPG produced by actively respiring tissue has and as a byproduct of glucose metabolism, the 2,3-BPG fits into what they call the whole of the donut of adult hemoglobin. I think I have that here. You can see it on the right, whole of the donut. So it fits into the whole of the donut, and this also favors the T state, or tight, low oxygen binding. Let's see, absence of 2,3-BPG, hemoglobin can more easily exit the T state and return to the R state. Smokers actually have higher concentrations of 2,3-BPG than non-smokers. So a lot more of their hemoglobin remains in the T state and thus carries less oxygen. So another reason actually why why smokers' oxygen carrying capacity is lower than non-smokers is that when they're smoking cigarettes, cigarette smoke has carbon monoxide in it. This molecule can actually go and outcompete oxygen for binding with the heme in hemoglobin. So then again, not carrying as much oxygen around. And that's part of why carbon monoxide is so toxic, actually, is because it just outcompetes hemoglobin. So you can kind of see, I thought this was a carbon monoxide. Yeah, this is carbon monoxide. So you can see carbon monoxide and oxygen, molecular oxygen, almost have like the same dimensions. It's just a much tighter bond. What was I showing about this too? Oh, this is important too. So when we're talking about the Bohr effect, we're talking about how these are all coupled together. And so you can see here, when actively respiring tissue, meaning when we get to glycolysis and citric acid cycle, we're going to see that molecules of CO2 are breathed out. Now, CO2 isn't soluble, very, very soluble in aqueous solution. And so it's going to react with water, okay, to form, I think this is bicarbonate, what is this? H2, H2CO3, carbonic acid. So this forms carbonic acid. And then carbonic acid actually exists in equilibrium with bicarbonate, HCO3 minus. Well, when this happens, right, we see that hydrogen ions are released. So these are all tied together. So increases in carbon dioxide lead to increases in hydrogen ion. And these are going to affect the structure of the what is the word I'm looking for? They're going to affect the structure of the the hemoglobin. Your book goes into a little more detail as well, how all of these different forms, the CO2, not solid, aqueous, and the CO2 gas are in equilibrium with each other, and that as we breathe out CO2 gas from the lungs, concentration becomes low, and so that shifts a lot of these equilibrium reactions here back and forth. We'll talk a little bit. I think we'll talk a little bit about this more later. But thinking back to the lecture that we had about buffers, and I'm hoping that you guys read the chapter on that. the book talks about the two kinds of buffers that we have, and one of those is the bicarbonate buffer that we actually have in our blood, and then we have a phosphate buffer actually that we use in our cells. Anyways, so just thought I'd throw that in there. One of the differences actually for babies in one of the reasons that they can actually prioritize, or the mom's body actually prioritizes the baby getting more blood, is that for a few reasons actually, more oxygen I should say. So adult hemoglobin releases oxygen when it binds 2,3-BPG. In contrast, fetal hemoglobin, which has a slightly different configuration than adult hemoglobin, it has 2 alpha chains and 2 gamma chains, so it doesn't have 2 beta chains. This actually has a greater affinity for oxygen than the mom's hemoglobin, than adult hemoglobin. So it can already get oxygen better than the mom can. Secondly, fetal hemoglobin doesn't bind 2,3-BPG. So the differences in those chains, in those subunits of the quaternary structure, actually prevented binding a molecule that lowers its affinity for oxygen and also has built into it an increased affinity for oxygen. It's pretty cool. I think I've got that image here. What was I going to show? This is sickle cell anemia. This is what happens to red blood cells with one amino acid difference, amino acid mutation or substitution at a specific part in the sequence leads to misfolding of the proteins. We talked about some misfolded proteins in the last lecture. Sickle cell anemia leads to this characteristic kind of sickle shape, which can make these blood cells very fragile. They get stuck passing through arteries. They break. So diminished blood carrying or oxygen carrying capacity. Sorry. So again, hemoglobin or fetal hemoglobin, higher affinity for oxygen than adult hemoglobin. And you can even see how that changes the amount of, let's see, so we're looking at in this table here, as the baby ages, the amount of the types of hemoglobin that they make. And so initially they make lots of A. Everybody needs alpha. They initially start off making lots of gamma and not a lot of beta. And then at about a couple weeks after birth, so between 4 and 5 weeks after birth, that kind of inverts. So actually even before then it starts to happen. The gamma production goes down, beta production goes up. Because once they're breathing on their own, they don't need this gamma one anymore. And you might think, why would you ever want to carry less oxygen? Well, it's not about carrying less, it's about being able to regulate it. Gamma chains don't allow for 2,3-BPG binding, and 2,3-BPG is a way of regulating that enzyme, that oxygen carrying protein. So we want that. We want to regulate stuff. Your body does. Okay. So yeah, sickle cell. What else? What was I going to talk about? Let's see. Oh. All right. So this is a myoglobin. You can see myoglobin here actually bound to oxygen. So the oxygen molecule is here. The heme group is there. You can see all of these secondary structures like alpha helices, intrinsically disordered, what did we call those? Random portions of the protein. So myoglobin is the primary oxygen storage protein found in animal muscle tissue. In contrast to hemoglobin, which circulates through the body, myoglobin is only found in muscle tissues and appears in the blood only after injury. Like hemoglobin, it has a prosthetic heme group that contains the iron. Now, the red color of meat comes from the heme of myoglobin, and the browning of meat by cooking it comes from the oxidation of that iron 2+, that ferrous iron inside of the heme group, and it oxidizes into iron 3+. So oxidation, remember the loss of electrons. So in the cooking process, that's what happens, and that's why meat turns the color it does. Now, as meat sits out in our atmosphere, right, in the oxidation or the oxygen-rich environment, it also turns brown because of the same reason, iron 2+ turning to iron 3+. If it's stored in carbon monoxide in a CO environment, then the carbon monoxide actually binds to the heme group and reduces the amount of oxidation, keeping the meat looking red for a longer period of time. So they actually inject carbon monoxide into meat at the packet, in the packages, so that it looks really beautiful and red. Now, myoglobin displays higher affinity for oxygen at low oxygen concentrations than hemoglobin. We talked about that. And therefore, it's able to absorb oxygen delivered by the hemoglobin under those conditions. So, hemoglobin takes it to the tissues, releases it, myoglobin grabs it. The protein exists as a single subunit in contrast to those 4 subunits. And let's see, mammals that dive really deeply in the ocean, like whales and seals, they have muscles with really high abundances of myoglobin. And it makes sense. When oxygen concentrations in the muscles falls really low, myoglobin releases its oxygen. So it's kind of like an oxygen battery that delivers oxygen when you need it. Interestingly, and this is just what it looks like here, what the oxygen bound heme looks like. This is like the zoomed in this person, or this version. Myoglobin actually holds the distinction of being the first protein for which the 3D structure was determined by X-ray crystallography. This happened in 1958. So, we've got this beautiful structure. We gotta not take it for granted. It's been, what is it, like less than 70 years since we've been able to actually see these things using X-ray crystallography. So the field is really just getting ready to blow up, right? Because we can barely, only for the last 70 years, and not everybody has X-ray crystallography, at their disposal. So imagine going forward from today, how when these things become more available and we start to really see the structures of these proteins, what we're really going to understand and what we're going to know and how much we're going to learn. It's going to be so much cooler. Okay, anyways, that is, I think, the end of the slideshow. So, yeah, all right. So I think the next video that we're going to do, the next lecture topic is going to be enzymes. So stay tuned. See you guys next time.